Originally, cyclopeptide, this is our crystal structure of a cyclopeptide, one of the things we've recently published. It was shown to suppress immunity, and we believe that it might work through some mechanism of blocking cell division, possibly through apoptosis, and it does slow the growth and proliferation of T and B cells. It binds to human serum albumin. This is just showing the test that we've used. In essence, what we do is we have a flow of cyclopeptides. We make a chip which has human serum albumin on the chip, and we flow the peptides by, and we can actually see the difference in the refractive index, and thereby measure how much binding is going on. So when you're doing a study like this, you have this is the chip with the human serum albumin bound to it, and the peptides would flow by, and you would see an absorption peak. And then if you started washing water over the chip, you could see the peptides being released and the like. And this kind of test actually tells you whether the peptide is going to be drug-like in certain applications. So you'd be looking at the peptides with the intention of finding out, are they going to move in the body, or are they going to be released in different parts of the body when you use them. This is actual data of cyclopeptides binding and releasing from human serum albumin. You can see the light changing in direction as the peptide binds to the chip. We work in a College of Agriculture, and there's a lot of people doing animal feeding trials through there. And there's also I have associates within Agriculture and Agri-Food Canada who've been doing feeding trials with flax. And what we wanted to do was look at cyclopeptides in these animals after they've been consuming flax products. What we found is that with pigs, as you see in this diagram here, cyclopeptides A and C are found in the fat. So this is done in collaboration with Mike Dugan at Lacombe. And we also found that it occurred in trout oil after they were fed flax oil. So the cyclopeptides A and C are getting into the animal tissues and are actually present in the tissues after they eat the materials. We also looked at the presence of a heat shock response in nematodes. We were actually growing nematodes in small petri dishes and looking at what happens when we put cyclopeptides on them in their response. Now, needless to say, they actually die. But those that we feed very low concentrations, if we look at them, this would be a control and a DMSO treated, because the cyclopeptides are quite hydrophobic, so we have to use DMSO to get them into solution. And as we look at very low concentrations of the peptides, we can see that at very low concentrations, the peptides are inducing the production of heat shock protein. And when the concentrations get higher, they produce less heat shock protein. Of course, they're dead, which might explain they probably just died really fast and couldn't express any proteins. But that's one of the things that we've noticed as well. There's a lot of diseases that are related to nematode infections. Human lung epithelial cancer cell line, we've also looked at the impact of them on these types of cell lines. And we looked at in QRT, PCR, and the expression of a gene that's associated with apoptosis after cyclopeptide A treatment. And you can see this is the control, DMSO levels. And as we put more and more cyclopeptide, we're seeing a lower and lower response. But at very low responses, we're seeing a very strong response to the cyclopeptide in the induction, similar to camptothecin, which is a known inducer of apoptosis.